This is the nuclear envelope, and it has two membranes. So it was once a swallowed bacterium. No. And that one fact is where most of the marks on this topic go. The nucleus fails every other test. No circular genome, no bacterial ribosomes, no dividing on its own. Two different origin stories built this cell, and telling them apart takes 4 pieces of evidence. Start with the simple cell. One membrane on the outside and no compartments at all inside. Story 1. The outer membrane pushes inward, folds, and just keeps going. Those folds become the endoplasmic reticulum, the Golgi, and the envelope around the nucleus. And notice what a fold gives you for free. Push one membrane in, and you get 2 layers automatically. That is the whole infolding story. But 2 organelles are not on that list. Story 2 starts outside the cell, with a free-living bacterium that breathes oxygen. The host cell reaches around it. This is engulfing, the same move it uses to eat. The membrane closes behind it, and the bacterium ends up inside, wrapped in a piece of the host. And here's the twist that made us. It is not digested. It survives in there, and it stays. Count the membranes now. The inner one is the bacterium's own. The outer one is the host's. So here's how you identify one. 4 fingerprints. We just watched the first one form. Second, it carries its own DNA, and the shape is the evidence. A loop, like a bacterium's. Third, its own ribosomes, and they're the small bacterial kind. The cell's own are bigger. Fourth, it divides by pinching in 2 on its own schedule, whatever the cell around it is doing. 4 for 4. That is not a membrane fold. That is something that used to be a cell. The chloroplast scores the same 4. Same story, a different bacterium. Now the case that decides whether you actually understand this. The nuclear envelope. It has 2 membranes. On the first fingerprint, it looks like a perfect match. But it has no genome of its own. No bacterial ribosomes, and it never divides by itself. 1 out of 4. The nuclear envelope came from a fold, and its 2 layers are what folds do. Which is the whole lesson. One fingerprint proves nothing. Endosymbiosis takes the entire set. There were 2 engulfments, not 1. The first took in a bacterium that could use oxygen. That one became the mitochondrion, and every eukaryote alive is descended from that cell. Much later, a second engulfment took in a bacterium that could photosynthesize. That became the chloroplast, and only the plant and algal line ever got it. Animals never did. So keep 2 buckets. Infolding built this side, engulfment built the other, and only these 2. The classic overreach is stretching infolding to cover everything, including these 2. It runs the other way too. Nothing engulfed the endoplasmic reticulum. 2 origins, and each explains exactly its own half of the cell. Do not mix them. On the exam, this comes as justify. That word is asking you for the evidence, not the story. So do not hand back one fact. Hand back all 4, and say what each one rules out. Circular DNA, bacterial ribosomes, a double membrane, and division by fission. That is the answer. Want to step through this one yourself? Links below.